Thank you for your interest in our webinar, InCELL Target Engagement Assays for Drug Discovery. In this presentation, I will describe what cellular target engagement assays are, explain the principles and technology behind our InCELL target engagement assays, provide examples of their use in drug discovery, and end by discussing DiscoverX's products, services, and resources to support your cellular target engagement assay needs. Many in vitro assays performed in drug discovery can be classified as either target engagement or functional assays. Target engagement assays measure ligand binding to a target, here shown to be a receptor, but don't measure the downstream functions which result from ligand binding. Note, in this webinar, I will use the terms compound and ligand interchangeably. Functional assays measure downstream events that are initiated by ligand binding to their targets. Examples of functional assays for GPCRs are measurement of second messengers, such as cyclic AMP and calcium, and recruitment of beta-arrestin. For a receptor tyrosine kinase, examples of functional assays would be subunit dimerization and adapter recruitment. Unlike the receptors shown here, not all targets possess good functional readouts for development of assays. These targets are often still excellent targets for developing cellular target engagement assays. One other point to make is that unlike functional assays, target engagement assays are agnostic to the mechanism of action of the ligand. They can detect the binding of agonists, antagonists, and everything in between. Target engagement assays come in two general types, cell-free or biochemical and cell-based or cellular. For both cell-free and cellular target engagement assays, we get information about binding affinity to specific target proteins or sometimes multiple targets. We get this for one or more compounds. Analysis of multiple compounds allows generation of rank order of target engagement information, or rank order of binding, affinity, or potency. For cellular target engagement assays, we get all of the above information coming with a cellular context, and we also get information on the cell permeability for the compound or compounds. In other words, cell-based target engagement assays assess the combined properties of target engagement in the cell plus the cell permeability for the compound. The remainder of the webinar will focus on cellular target engagement assays.
There are multiple applications for cellular target engagement assays in drug discovery. Some of these applications include taking hits from biochemical assays and learning about cellular permeability and potency of the compounds, analyzing challenging targets, for example, where no intrinsic catalytic activity or straightforward assay exists, running primary screens of compound libraries in more relevant cellular assays, improving the relevance of assay data during lead optimization, and deconvolution of hits from phenotypic screens. In other words, identifying the responsible targets in a cellular environment. As shown in these schematics of target-specific and phenotypic drug discovery approaches, cellular target engagement assays, as well as functional assays, play important roles in both paradigms for drug discovery. Let's discuss how a cellular target engagement assay works. First of all, cellular target engagement assays take place in cell culture systems, which are comprised of cells in a liquid medium or buffer, typically in a tissue culture multi-well plate. The cells used for target engagement contain the target protein and all other cell proteins and structures, in this case in their cytoplasm. But cytoplasmic localization is not absolutely required. Next, a compound under study is added to the liquid medium or buffer bathing the cells, as shown in step 1. The compound then enters the cell's plasma membrane, which is a permeability barrier which compounds will need to cross to gain access to the cells. If the compound is cell permeable, it crosses the cell membrane as shown in step 2, enters the cells, and distributes into the cytoplasm and cell interior, as shown in step 3. Here, the compound encounters the target protein labeled 4A. If the compound is a ligand for the target and has affinity for the target, The compound will bind to the target protein as seen in step 4, resulting in a target with compound engaged or bound, labeled as 4b. The target protein exhibits different biological and biophysical properties without and with ligand bound. Specifically, the turnover and thermal stability usually is different without and with ligand bound. And this difference in biophysical and biological properties is what is measured in a cellular target engagement assay. DiscoverX offers 2 cellular target engagement assays, InCELL Pulse and InCELL Hunter. Both formats measure the cellular level of native, properly folded, user-defined target protein, but they approach this somewhat differently. InCELL Hunter relies on a target protein's normal dynamic equilibrium between synthesis, accumulation, and degradation in cells. For InCELL Hunter, compound target engagement is measured by a compound's ability to shift the steady state away from degradation and towards accumulation. InCELL Pulse relies on a target protein's tendency to denature at elevated temperatures. Plotted with black circles is the thermal denaturation or melting curve of user-defined target protein in the cells in the absence of compound binding. At physiological temperatures on the left, we have natively folded functional protein, giving a high signal in the assay. At higher temperatures, we have unfolding and then denaturation, giving rise to lower assay signal. In the InCELL Pulse assay, target engagement is measured as a compound's ability to increase the target protein's thermal stability in cells. Plotted with red squares is the thermal denaturation curve with a single high concentration of ligand bound to the target protein. If we look at 51 degrees centigrade, without compound, we see a loss of close to 90% of the target protein signal, whereas in the presence of bound compound, about 70% of the target protein signal remains. By binding, the compound has stabilized the target protein against thermal denaturation, resulting in more remaining functional protein. This difference in signal with and without bound ligand can be described as the assay window. The displacement of the red curve to the right to higher temperatures can be described as the TM shift or the thermal shift in the presence of compound binding. Importantly, while not shown here, at any given temperature, the amount of stabilization of the target protein is dependent on the concentration of the ligand compound added to the assay. This relationship allows the generation of isothermal dose-response curves reflective of a compound's potency for binding to the target in the cells. Eurofins DiscoverX's InCELL cellular target engagement assays are based on split beta-galactosidase enzyme fragment complementation technology, also known as EFC. EFC involves splitting beta-galactosidase into 2 inactive fragments, a smaller, approximately 40 amino acid tag referred to as enzyme donor, ED, or enhanced prolabel, EPL, and a fragment called enzyme acceptor, EA. Binding of ED to EA reconstitutes beta-galactosidase activity, which allows hydrolysis of added substrate, resulting in generation of a chemiluminescent assay signal. In InCELL target engagement assays, the target protein is tagged with the EPL tag. Addition of EA at the end of the assay detects increased levels of EPL tag target due to compound target engagement as increases in assay signal. DiscoverX offers 2 InCELL cellular target engagement assays, InCELL Hunter and InCELL Pulse.
Both assays start with the same cell line expressing an EPL-tagged user-defined target protein. Both assays use the same detection reagent, which contains enzyme acceptor, EA, and luminescent substrate. While not shown in this illustration, both assays also provide graphical data in the same format. For the InCELL Hunter assay, with compound binding, we have a stabilization of the EPL tag target protein in cells, making it more abundant and more available to complement with EA. And this results in more reconstituted beta-galactosidase enzyme and more luminescent assay signal. This assay relies on the physiological turnover of target protein in cells and generally requires a 7-hour workflow. In the InCELL pulse assay, a short pulse of elevated temperature denatures and aggregates the EPL target protein, here shown without compound binding. Compound binding stabilizes the EPL tag target protein against thermal denaturation in the cells, making it more abundant and more available for complementation of the enzyme acceptor. This results in more hydrolysis of substrate and more luminescent signal. This assay relies on a thermally accelerated denaturation of target protein and requires a 2-hour workflow.
Requirements and availability of assays for cellular target engagement. So what are the requirements for a cellular target engagement assay? They should be quantitative, sensitive, and accurate, easy to use and compatible with high throughput screening, low cost without the need for custom reagents or complex hardware, and broadly applicable to a wide range of targets. So how do currently available assays meet these needs? Most available assays are cumbersome and low throughput, involving antibodies for Western blot or ELISA, chemically modified compounds, or complex mass spectrometry-based readouts. On the other hand, Eurofins DiscoverX's InCELL Hunter and InCELL Pulse cellular target engagement assays are simple, rapid, homogeneous assay methods with simple readouts using a standard laboratory plate reader. No specialty reagents or complex hardware are required, and they allow measurement of compound cell entry and target interaction, allow validation of hits from biochemical and phenotypic screens, and can be used to confirm a compound's mechanism of action, and overall provide a powerful solution when good cell-based functional assays are difficult or unavailable. The workflow for the InCELL Pulse target engagement assay is simple and rapid. This workflow involves adding compounds to a PCR plate and then adding cells expressing a user-defined EPL tag target protein. These first two steps can be performed in either order. Next, the compounds are incubated with the cells for 1 hour. A pulse denaturation step is performed, requiring 6 minutes. Next, detection reagent is added and incubated with the cells for 1 hour, followed by reading the chemiluminescent signal, analyzing and plotting the data. Here the data are shown as concentration of compound versus assay signal. This assay workflow takes 3 hours of time. While not shown here, the InCELL Hunter assay workflow is similar with 2 primary differences. First, the compound incubation step is 6 to 16 hours. And second, the thermal pulse denaturation step is absent. Importantly, the same user-defined target-expressing cell lines and detection reagents are used for both the InCELL Hunter and InCELL Pulse assays, and both assays provide the same insights into compound cellular permeability and target engagement using the same data format. So depending which assay works best for your target, either of the 2 assay formats is available with the same cells and the same reagents. This table compares various attributes of the InCELL Hunter and InCELL Pulse assays. Again, the main differences are whether they are built upon physiological turnover or thermal denaturation of the target protein, and differences in compound incubation time. The choice between InCELL Hunter or InCELL Pulse assay is typically made empirically during assay development. The availability of these two InCELL cellular target engagement assay formats allows the study of more target proteins. In the next set of slides, I will show you some examples using the InCELL Hunter and InCELL Pulse cellular target engagement assays for different applications. Analyze challenging targets with InCELL target engagement assays. MTH1, a DNA-modifying enzyme, and G9A, a histone-modifying enzyme, are both oncology targets without good functional assays. These curves show analysis of 3 MTH1 inhibitors and also a lipid kinase inhibitor, which is inactive, using the InCELL Pulse assay. The rank order of potencies of these 3 compounds are generally in good agreement with published values. On the right here, these curves show analysis of 3 G9A inhibitors and again the inactive lipid kinase inhibitor. In the next example, I'd like to drill down on additional studies with this target. These are more data from the G9 DNA hydrolase. In a biochemical assay, the potency of UNC0638 and UNC0321 were equivalent and much more potent than BIX1294. However, in the cell-based target engagement assay, UNC0638 was more potent than BIX1294 and much more potent than UNC0321. And here we're plotting the biochemical over cellular potency ratio for these 3 compounds. Thus, InCELL target engagement assays give information about cell potency and permeability of compounds previously tested in biochemical assays. InCELL target engagement assays indicate compounds with similar biochemical IC50s do not have the same potency for the target in a cell-based target engagement assay. Specifically, the cellular target engagement assay has revealed that compound UNC0321 may have reduced cell permeability. Understand potencies and specificities of kinase inhibitors using InCELL target engagement assays. These experiments each evaluated 4 inhibitors on either of 2 protein kinases, HCK, a Sarc family kinase on the left, and MEK1 on the right, using InCELL Hunter cellular target engagement assays. For both of these data sets, we can see good rank order of potencies of the different inhibitors, and for both of these data sets, the rank order of potencies obtained for the InCELL Hunter assay was in good agreement with published cellular potencies from the literature. This experiment further demonstrates how protein kinase target specificity and rank order of cellular potency of a series of kinase inhibitors can be evaluated using the InCELL Hunter target engagement assay platform. Here we see that PD35901 and U0126 are active against MEK1 in the cellular target engagement assay, with the PD compound being an order of magnitude more potent, while dasatinib is inactive. It is also interesting to note that the 2 active MEK1 inhibitors have different mechanisms of action, with U0126 binding to the active site, while PD35901 binds to an allosteric site. Of these 3 compounds, only dasatinib is active in the B lymphocyte kinase target engagement assay, and this compound was inactive against MEK1. Practically speaking, this demonstrates the absolute target specificity of these 3 inhibitors for either MEK1 or BLK, respectively. This is consistent with the specificities of these inhibitors from the literature, and also serves to validate the specificity of the InCELL assay platform. As shown in the previous slide, inhibitors with different mechanisms of action can be studied in the same InCELL cellular target engagement assay. Here is another related example. ABL1 kinase inhibitors with 3 different mechanisms of action can be studied in the ABL1 InCELL Pulse assay. The graph on the left shows dose-response curves for dasatinib and imatinib,
two active site binding inhibitors, which bind, however, to different conformations of the active site, called DFG in and DFG out, respectively. The graph on the right shows a dose curve for an allosteric site inhibitor, GNF2, in the ABL1 InCELL pulse assay. I should point out that studying competitive inhibitors in a functional assay requires stimulation by an agonist along with an antagonist. This, however, is not necessary in the InCELL target engagement assays. In contrast, when using InCELL target engagement assays, antagonists can be studied directly without need for use of an agonist as well. So in conclusion, it is important to recognize that InCELL target engagement assays measure ligand binding and are generally agnostic to a compound's mechanism of action. So, competitive inhibitor binding to an active site, allosteric inhibitors, and even agonists for a user-defined target can all be studied directly using a single InCELL target engagement assay. PROTACs are an important novel class of heterobifunctional compounds that bring targets and E3 ligases together, promoting target degradation, as shown in the cartoon at the top right. Targets for protec development are typically challenging drug discovery targets. For example, BRD4 has no readily assayable enzyme activity. BRD4 degrading protec MZ1 is comprised of BRD4 binder JQ1 and VHL binder VH298. This curve here shows a BRD4 InCELL target engagement assay with compound JQ1, and this curve over here shows a VHL InCELL assay with compound VH298. As shown, InCELL target engagement assays allow study of ligands for challenging protec targets such as BRD4 and E3 ligases such as VHL. Just to follow up on the previous example, we have actually shown that the InCELL target engagement assay signal, due to target stabilization, correlates with other external measures of target stabilization. In this example, sister wells to those circled here in this InCELL target engagement assay were analyzed by BRD4 immunoblot and showed comparable JQ1 dose-dependent increases in BRD4 as seen in the InCELL target engagement assay. These and other data from other experiments using orthogonal assays have served to validate the results of the InCELL target engagement assays and can also be run in your laboratory to validate your InCELL target engagement assays. InCELL target engagement assays can be extended to many protein kinase targets. In fact, we have developed InCELL target engagement assays against protein kinases across the human kinome. And InCELL assays are not just for protein kinases. We have a reference set of validated InCELL pulse kinase assays, 25 of which are shown here. Kinase validation data and reagents are available from discoverx.com/InCELL. InCELL target engagement assays have been successfully developed against a broad range of protein targets, as shown in this list, including enzymes, kinases, proteases, hydrolases, and ligases, protec targets, epigenetic targets, including bromodomain BET proteins, methyltransferases for nucleic acid and protein. This has included targets which are plasma membrane proteins, both single span and polytopic membrane proteins, and even organellar proteins. At DiscoverX, we are making an effort to cover all bromoproteins. Here are a few more examples of the more than 30 bromodomain protein InCELL assay cell lines we have available. So InCELL assays let you cover proteins from many classes and allow coverage deep within a protein class of interest. Owing to the simple workflow and excellent data quality,
InCELL pulse assays are highly amenable to high-throughput compound profiling and library screening. These data show high-throughput dose-response profiling of 123 compounds against a SMID3 histone lysine methyltransferase InCELL pulse assay in a 384 well plate format as performed by GSK scientists. Here are shown dose-response curves for 4 compounds. And here you can see excellent CV values for most of the compounds with high affinity in the assay. Numerous studies have been published by pharma drug discovery groups using InCELL target engagement assays, a sampling of which are shown in this slide. Links to all of these and additional references and resources are available at discoverx.com/InCELL. In summary, InCELL cellular target engagement assays are simple and rapid and allow determining cellular permeability and potency of compounds binding to targets in a cellular context, allow follow-up screening on biochemical hits, allow target deconvolution from hits from phenotypic assays, and allow analyzing multiple and challenging targets. Eurofins DiscoverX offers cell lines, ready-to-use kits, and starter kits for you to develop your own InCELL assays. We also offer InCELL cellular target engagement assay screening as a service and provide custom assay development of InCELL cellular target engagement assays. For more information, visit discoverx.com/InCELL.